Welcome to the Deep Dive, your express lane to becoming truly well-informed. Today, we're going to pull back the curtain on, well, the grandest show on Earth, life itself. Just pause for a moment and consider the sheer complexity, the elegant dance happening all around us and even within us. From the microscopic machinery of a single cell to these vast, sprawling ecosystems, it's this incredibly intricate ballet of systems and structures all working in concert. Most of the time, anyway.
It's a truly humbling thought, isn't it? What's so captivating about this field is just how deeply interconnected every single biological principle is, from the smallest molecule right up to the largest organism. For this deep dive, we're tapping into a really rich array of sources, everything from foundational biological notes to detailed descriptions of life processes and classifications. We're going to connect so many dots for you today, hopefully revealing some of the underlying logic of the living world.
Yeah, our mission is crystal clear. Navigate this immense ocean of biological information, pull out the most vital nuggets that explain how living things are structured, how they function, reproduce, and, maybe most importantly, how they interact with their environment. Get ready for those genuine aha moments, the kind that truly deepen your understanding of the incredible world around and within you.
Okay, let's unpack this intricate living tapestry. So when we talk about the animal kingdom, kingdom Animalia, what exactly are we defining? What makes an animal fundamentally an animal, sets it apart from, say, a plant or a fungus?
That's the perfect starting point. Fundamentally, animals are defined by, a few core non-negotiable traits. First, they are eukaryotic. That means their cells have a true nucleus and other membrane-bound organelles. It's a step up in complexity from, say, bacteria.
Okay, complex cells, got it.
Second, they are multicellular. They're composed of many specialized cells working together, forming tissues, organs, systems, the whole shebang. And third, and this is really critical, they are heterotrophic.
Heterotrophic, meaning they can't produce their own food like plants do through photosynthesis. They must get nutrients by consuming other organisms. Think of it as being a consumer in the grand biological supermarket.
Right. So no making your own snacks, you have to go out and find them. What else sets them apart?
Another key difference, especially compared to plants or fungi, is the absence of a rigid cell wall. This gives animal cells more flexibility, which ties into another key point.
Yeah.
Mobility. Most animals, at least at some stage of their life cycle, are generally mobile. That ability to move around, chase prey or escape predators, that's a defining characteristic that shapes so much of animal evolution.
That mobility often links directly to how their bodies are organized, doesn't it? Like symmetry. Biologists often look at body symmetry first. You see everything from totally irregular shapes to very organized structures. How do we break those down?
You're absolutely right. Body symmetry tells us a huge amount about an animal's lifestyle, how it interacts with its environment. At the simplest end, you've got organisms with no symmetry at all, or asymmetry. Imagine trying to cut a blob into two identical halves. You just can't, no matter how you slice it. This is typical of porphyra, the sponges.
The sponges, right, just kind of there.
Exactly. And that seemingly simple body plan is perfect for their lifestyle. They're stationary filter feeders. They just sit there, pulling water through their bodies from all directions. No need for a front or back. Then you have radial symmetry. This is a step up in organization. Animals like Coelenterata, which are also called Cnidaria jellyfish, corals, anemones, and also Echinodermata and Pseudonaphora, they display this. With radial symmetry, you can divide the animal into equal mirror image parts along any plane passing through its central axis. Think about slicing a pie.
Okay, like a pie or a wheel.
Precisely. And this design is ideal for animals that are either stationary, like a sea anemone, or drift slowly, like a jellyfish. It lets them sense their surroundings and grab food from all directions equally well. They don't really have a head end.
And then there's us, humans, dogs, fish. We don't fit those, do we?
Nope. That brings us to bilateral symmetry. This is what you and I have, along with the vast majority of the animal kingdom. We're talking worms like Platyhelminthes and nematodes, segmented worms like Annelida, insects and spiders and Arthropoda, and of course, all chordata. If an organism has bilateral symmetry, you can only divide it into two mirror image halves, a left and a right side along one single plane, the sagittal plane running down the middle.
Just one way to get two matching sides.
Exactly. And this body plan is incredibly efficient for directed forward movement. It allows for what we call cephalization, the concentration of specialized sensory organs like eyes, ears, and eventually a brain at the anterior or front end, the end that leads the way.
Which makes perfect sense if you're actively moving around, hunting, or trying not to be hunted.
It's a huge evolutionary advantage for navigating complex environments effectively.
Okay, so that's the overall shape. Moving deeper, animal development starts with layers, these germ layers. Why are they so crucial? They sound like blueprints.
They absolutely are the initial blueprints. Germ layers are fundamental embryonic tissues. They're the master construction plans from which all the different tissues and organs in an animal's body will eventually arise. Their presence, or absence, marks a really monumental evolutionary divide.
So how many layers can there be?
There are two main categories based on the number of layers. First, you have diploblastic animals. These guys, like porifera, sponges, coelenterata, jellyfish, corals, and ctenophora, comb jellies, develop from just two germ layers. There's the ectoderm, which forms the outer covering, like skin and nerves, and the endoderm, which forms the inner lining, like the digestive tract.
Just 2 layers. Simple.
Pretty simple. And interestingly, between these two layers, diploblastic animals often have a non-cellular, jelly-like matrix called mesoglia. It's not a true third layer with cells. This inherently limits their internal complexity.
Okay, so what's the other category?
That would be triploblastic animals, and this includes the overwhelming majority of the animal kingdom, everything from flatworms right up to insects, fish, and mammals. They develop from three germ layers. They have the ectoderm and endoderm like the diploblasts, but crucially they have a third layer, the mesoderm, sandwiched right in between.
And this mesoderm is the game changer.
Absolutely. The mesoderm is incredibly significant. It gives rise to a truly astonishing array of structures. All types of muscles, bones, the circulatory system, the excretory system, and perhaps most importantly, it gives rise to the true body cavity, the coelom. This addition of a third layer literally unlocked the potential for the sophisticated organ systems and complex body plans we see in more advanced animals. It's why you have a heart, kidneys, a skeleton, while a jellyfish is essentially, well, a simple bag.
Ah, the coelom. Okay, that brings us neatly to body cavities. Sounds like important internal real estate. What exactly is a coelom, and why does having one or not having one matter so much?
The coelom is basically the true body cavity. Imagine a fluid-filled space within your body positioned between your outer body wall and your internal gut wall. The critical defining feature of a true coelom is that it is entirely lined by tissue derived from that middle layer, the mesoderm.
Fully lined by mesoderm.
All right.
Yeah.
And its presence is a game changer. It provides several profound advantages. It allows internal organ space to grow, specialize, and move independently of the body wall. It offers cushioning. It allows for complex organ systems to develop and function efficiently without being squished. It's like upgrading from a cramped studio apartment to a spacious house with dedicated rooms for everything.
Makes sense. So how do we categorize animals based on this coelom?
We group them into three fundamental types based on the column. First, echolomates. The A means without. These animals, like platyhelminthes, flatworms, completely lack any true internal body cavity. Their bodies are basically solid between the gut and the outer wall.
Okay, no cavity. Flatworms.
Then you have the pseudocoelomates. Pseudo means false. Animals like nematoda, or achelminthes, roundworms, have a body cavity, yes, but it's a false cavity because it's not fully lined by mesoderm. It's like a partially finished room of their space, but not properly lined.
Got it. A sort of cavity.
Exactly. And finally, we have the eucoelomates. E means true. These are the animals with a true coelom fully lined by mesoderm. This group includes annelida, segmented worms, arthropoda insects, spiders, mollusca, snails, clams, echinodermata, starfish, and all chordata, including us. It's in these eucoelomates that you see highly developed, well-accommodated, independently functioning organ systems. That true coelom really allows for complexity.
So the coelom isn't just empty space. It's organized space that enables complexity. Okay. Beyond that, we also classify animals by things like their circulatory systems, whether they have a notochord, and segmentation. Can you break those down for us?
Absolutely. Let's start with circulatory systems. These are vital for transport moving nutrients, gases, hormones, waste products all around the body. There are two main types. An open circulatory system is one where blood isn't always confined within vessels. Instead, it flows freely through large body cavities, directly bathing the organs. This is common in many invertebrates, like arthropoda. Their body cavity, the hemocoel, is basically filled with blood, or hemolymph. It's generally less efficient for rapid transport over long distances.
Okay, so blood just kind of sloshes around.
In a way, yes. In contrast, a closed circulatory system means the blood is always enclosed within a network of vessels, arteries, veins, capillaries, and pumped by a heart. It circulates in a continuous, contained loop. We as humans have this, and so do all chordata. This closed system allows for much more efficient, rapid, and controlled transport, which is essential for larger, more metabolically active animals.
And the notochord, that sounds foundational, especially given our phylum name, Chordata.
It is absolutely foundational. The notochord is a flexible, rod-like structure that runs along the dorsal or backside of an animal during its development. It's derived from the mesoderm. Its presence, at least at some point in the life cycle, is the defining characteristic for all members of the phylum Chordata. It provides crucial skeletal support. In vertebrates like us, this notochord is largely replaced by the bony backbone, the vertebral column, during embryonic development. But its presence early on is that key evolutionary marker.
Gotcha. And lastly, segmentation, like an earthworm's rings.
Exactly like an earthworm's rings. Segmentation refers to the division of the body into repeating units or segments. Think of a train made of similar carriages. This feature is really prominent in phyla like Annelida, earthworms, Arthropoda, think of an insect's head, thorax, and abdomen segments. And even in Chordata, you can see remnants of it in the repeating pattern of our vertebrae and muscles. Segmentation is a fantastic evolutionary innovation. It allows for specialization. Different segments can become modified for different functions. It provides flexibility and allows for more efficient movement. It's like a modular design for building bodies.
That's a fantastic overview of the big picture classifications. Very clear. Now, let's maybe take a quick journey through some of the specific animal phyla, hitting the highlights, key characteristics, examples. We'll start simple and build up. First up, Porifera, sponges. What should we remember about them?
Porifera, the sponges. They represent the simplest form of multicellular animal life. Really basic. Their key feature is a body perforated with thousands of tiny pores called ostia. These lead into a central cavity, the spongocoel. This creates a water canal system. Water comes in the ostia, they filter out food, water goes out. Simple, but effective filter feeding. They have asymmetrical bodies, no way to cut them into equal halves. And they operate at a cellular level. Organization cells work together, but no true tissues or organs.
So just a collection of specialized cells?
Pretty much. They are diploblastic, 2 germ layers, exclusively aquatic, mostly marine, but some freshwater, and they're non-modal. As adults, they anchor themselves. Many are also hermaphroditic, having both male and female parts. Examples: Cecon, Spongilla, the freshwater one, Euspongia, the bath sponge. Shows how simplicity can still be successful.
Okay, from static sponges to colinterata or cnidaria, jellyfish, corals, what evolutionary steps do we see here?
Cnidarians represent the first group with true tissue-level organization. Cells are starting to form functional groups. They have radial symmetry and are diploblastic. Many, like corals, build hard exoskeletons of calcium carbonate, forming massive reef structures. Huge ecological importance there. They have a central gastrovascular cavity with a mouth surrounded by tentacles. But interestingly, no anus. It's a single opening for both intake and waste.
A two-way street, huh?
Exactly. But their most iconic feature, those specialized stinging cells called nematocysts. They're housed in cells called cnidoblasts. Think of them as tiny, spring-loaded harpoons used for defense and capturing prey. Even a simple jellyfish has these incredible microscopic weapons. Examples include corals, hydra, sea anemones, and jellyfish.
Wow. Okay, next, Ctenophora, the comb jellies. They sound kind of ethereal.
They really are quite stunning. Ctenophores are exclusively marine. They also have radial symmetry, but their defining feature is how they move using 8 rows of fused cilia that look like combs, beating in waves. And they are famous for their bioluminescence. They can produce their own light, glowing eerily in the dark ocean depths. Also hermaphroditic, reproducing sexually. You might see pleurobrachia or Ctenoplana.
Cool. Okay, now we're moving firmly into the world of worms. Let's start with Platyhelminthes, the flatworms.
Platyhelminthes, aptly named. Their bodies are dorsiventrally flattened like a ribbon or a leaf. This phylum marks some significant evolutionary steps. Bilateral symmetry, organ-level organization, tissues forming specific organs, and they're triploblastic, all 3 germ layers present. However, critically, they are a chelomate. They lack that true internal body cavity. They're solid inside.
Oh, so triploblastic but no coelom yet.
Correct. They also have an incomplete digestive system, just one opening. And for excretion, they have unique specialized cells called flame cells. Many are parasitic, like tapeworms and liver flukes, often having hooks and suckers to attach to hosts. But some, like planaria, are free-living, typically hermaphroditic.
Okay, flatworms. Then the roundworms, nematodes, also called Aschelminthes.
Yes, pneumatodes.
Yeah.
Distinctive, cylindrical, unsegmented body, hence roundworms. They also have bilateral symmetry, are triploblastic, and show organ-level organization. But here's where the body cavity gets interesting. They have a pseudocarpellum, that false cavity, not fully lined by mesoderm, an intermediate step.
Right, the sort of cavity again?
Exactly. Unlike flatworms, nematodes have a complete alimentary canal, separate mouth and anus, more efficient digestion. And significantly, their sexes are separate. They're unisexual, male or female individuals. Many are parasitic and pathogenic. Think of Ascaris, intestinal roundworm, filarial worms causing elephantiasis, pinworms, hookworms, and cyclostomas. They cause a lot of human diseases, but they're also incredibly important in soil ecosystems.
Then we get to Annelida, the segmented worms, earthworms, leeches. What's really special about them?
Annelids are where we see true segmentation become really prominent, both externally and internally. They're bilaterally symmetrical, triploblastic, and importantly, they are the first group we've hit that are true collimates. They have that genuine mesoderm-lined body cavity.
Finally, a true column.
Yes. And this allows for more complex internal structures and organ system organization. Big step up. They can show sexual dimorphism, distinct male-female forms. Some marine ones, like Nereis, have paired lateral appendages called parapodia for swimming or crawling. Their digestive system is a complete tube-like alimentary canal. Earthworms and leeches are the most familiar examples. They've colonized freshwater, marine, and terrestrial habitats, showing the success of that segmented plan.
Okay, now for the heavyweight champion of the animal kingdom in terms of numbers, Arthropoda. Insects, spiders, crustaceans.
Ugh. Arthropods. This phylum truly dominates. Billions and billions of them. Defining features, instantly recognizable. Jointed legs. Arthropod means jointed foot. A segmented body, usually clearly divided into head, thorax, and abdomen, or sometimes a fused cephalothorax. Bilaterally symmetrical, triploblastic. But they have an open circulatory system. Remember, blood sloshing around in the hemical.
Open system.
Respiration varies. Aquatic ones, like crabs, use gills. Terrestrial insects use a network of air tubes called trachea that deliver oxygen directly to tissues. Excretion is often via Malpighian tubules, and they all have that tough exoskeleton made of chitin.
The crunchy outer shell?
Exactly. Provides protection, support, muscle attachment.
Yeah.
But they have to molt it to grow, which is a vulnerable time. Typically unisexual. This group is just massive. Honeybees, silkworms, mosquitoes, locusts, spiders, scorpions, crabs, crayfish, shrimp, even ancient things like the king crab. Their success is down to that adaptable body plan and exoskeleton.
Incredible diversity.
Okay.
Following arthropods, we have mollusca, the second largest phylum. Snails, octopuses, often soft-bodied.
Yes. Mollusks are known for their soft bodies, though many protect themselves with a hard exoskeleton or shell made of calcium carbonate, like snails or clams. They're bilaterally symmetrical, mostly, and possess a unique feature called a mantle, a fold of tissue that drapes over the body and usually secretes the shell. They also typically have a distinct muscular foot used for locomotion, although this is highly modified in different groups. Think of a snail's gliding foot versus an octopus's arms.
And blue blood, I heard that somewhere.
You did. Many mollusks have blue blood because their respiratory pigment is hemocyanin, which is copper-based, unlike our iron-based hemoglobin. They have a complete alimentary canal, kidney-like organs for excretion, and usually respire via gills. Generally unisexual. Huge variety here. Snails, slugs, mussels, oysters, clams, scallops, the super intelligent octopus, sometimes called devilfish, squids, cuttlefish, a really fascinating phylum.
Definitely. And then the spiny skin echinodermata, starfish, sea urchins, quite different looking.
Echinoderms are exclusively marine and instantly recognizable by their spiny skin. Echinoderm means spiny skin. What's really weird is that while their larvae have bilateral symmetry, the adults often develop radial symmetry, usually 5-part radial symmetry, like a starfish.
They change symmetry. Wow.
Yeah, it's quite unusual. They are triploblastic, have a true coalomic cavity and organ-level organization. But their most unique defining feature is their water vascular system. It's this complex network of hydraulic canals branching into little tube feet. They use it for movement, feeding, respiration, excretion. It's a biological marvel, truly.
Yeah.
Starfish, Assyrias, sea urchins, echinus, brittle stars, sea lilies, sea cucumbers all show this unique system.
Okay, and finally, the phylum we belong to, Chordata. What unites such diverse creatures, from a tiny fish to a blue whale to us?
Chordates are unified by a set of 4 key features that appear at some point during their life cycle, even if only in the embryo. These are 1, a notochord, that flexible dorsal rod, 2, a dorsal hollow nerve cord, 3, paired pharyngeal gill slits, we have them as embryos, and 4, a postanal tail. They also exhibit bilateral symmetry, are triploblastic, have an organ system level of organization, a closed blood vascular system, and typically a ventral heart on the belly side.
So those 4 features are the key identifiers, even if temporary.
Exactly. Chordata is then broadly divided. You have the simpler protochordata, like sea squirts herdmania and lancelets amphioxus. In these, the notochord might not persist throughout life, but they have the key chordae features. They're marine filter feeders mostly.
And then the group we're in, the vertebrates.
Right. Vertebrata is where the notochord is properly developed into or replaced by a segmented backbone, the vertebral column. This provides much more robust support. Within vertebrata, we see several distinct classes, each with incredible adaptations.
Let's quickly run through those classes.
Okay, first the most primitive. Cyclostomata, the jawless vertebrates, think lampreys, Pittermyzon, and hagfish, Maxine. Eel-like, often parasitic or scavenging, no true jaws. Then Pisces, the fishes, a huge, diverse group of aquatic vertebrates, key feature. A two-chambered heart. We divide them into cartilaginous fish, skeletons of cartilage like sharks, rays, skates, seedy, dogfish, and bony fish, skeletons of bone, the vast majority of fish, tuna, rohu, catfish, salmon, trout. Also includes weird forms like the flying fish and the seahorse.
And the ones that made the leap to land, partially?
The amphibians. Their name means dual life, adapted for both water, usually for reproduction, larval stage, and land. They're cold-blooded, ectothermic, typically have a three-chambered heart. Their skin is moist with mucus glands, allowing for skin respiration, supplementing their often simple lungs. Tadpoles have gills. Think frogs, toads, salamanders, newts.
Okay, then the reptiles, more fully terrestrial.
Generally, yes. Reptiles are also cold-blooded and usually have a three-chambered heart. Big exception. Crocodiles, which uniquely have a four-chambered heart, like birds and mammals. Their bodies are covered with protective scales to prevent water loss, and crucially, they lay amniotic eggs, often with leathery shells, on land, freeing them from water for reproduction. Snakes, lizards, turtles, tortoises, crocodiles, alligators, all reptiles.
And the masters of the sky, Oves the birds.
Ives the birds, marvels of adaptation for flight. They're warm-blooded, endothermic, maintaining a constant body temperature. They have a highly efficient four-chambered heart, crucial for the high metabolic demands of flight. Their bones are typically light and often hollow. They have beaks instead of teeth, and of course wings covered in feathers. They're oviparous, laying hard-shelled amniotic eggs, usually cared for in nests. Crows, pigeons, eagles, penguins, flightless swimmers, ostrich, flightless runners, peacock, incredible diversity. And finally, we arrive at mammalia, the mammals, our own class.
What defines us mammals?
Mammals, like birds, are warm-blooded and have that efficient four-chambered heart. Our bodies are typically covered with hair or fur, providing insulation. But the absolute defining characteristic, the reason for the name, is the presence of mammary glands in females, which produce milk to nourish their young. Most mammals are viviparous, giving live birth. But there are fascinating exceptions. The monotremes, like the platypus and echidna in Australia, are unique egg-laying mammals. Shows evolution's creativity. Otherwise, think whales, dolphins, bats, elephants, kangaroos, monkeys, apes, and humans, a hugely successful and diverse class.
What an absolutely incredible journey through the animal kingdom's diversity. Okay, let's pivot now to one of the most fundamental processes in all of biology, reproduction. It's often said, isn't it, that reproduction isn't strictly necessary for an individual's survival, but it's absolutely crucial for the species. Can you unpack that idea?
That's a really profound way to put it, and it's absolutely true. Think about it. An individual organism needs to eat, breathe, get rid of waste, maybe move around just to sustain itself day to day. But reproduction, the whole complex business of creating offspring, isn't about that individual's immediate survival. It's about something much bigger, the perpetuation of the species across time, across generations. Without reproduction, even the healthiest, longest-lived individual would eventually die, and its lineage, its kind, would just vanish. So reproduction is really the ultimate act of biological legacy. It ensures the continuity of life. It allows species to adapt, to evolve, to persist in a world that's always changing. It's really the engine driving evolution.
And there are two main fundamentally different strategies, right? Asexual versus sexual reproduction. What are the core differences and maybe the pros and cons of each?
The fundamental difference really boils down to the number of parents involved and, crucially, the genetic outcome for the offspring. Asexual reproduction involves just a single parent. It's incredibly common in simpler organisms, lots of bacteria, protists, even some plants and simpler animals. The key result, offspring that are genetically identical to the parent. Essentially, perfect clones.
Like making copies.
Exactly. This strategy is fast, it's efficient, you don't need to find a mate, you don't need complex reproductive organs. Great for rapidly colonizing a stable environment. However, the big drawback is the lack of genetic diversity. If the environment changes, or a new disease pops up, the entire identical population could be wiped out.
Okay, so efficiency versus risk. What about sexual reproduction?
Sexual reproduction, on the other hand, typically involves two parents, usually a male and a female, contributing genetic material. This is the dominant mode in higher plants and most animals. Its defining characteristic is the combination of genes from two different individuals. This results in offspring with unique genetic combinations, different from either parent and from each other. This generates genetic diversity.
And that diversity is the key advantage.
Absolutely. Genetic diversity is the raw material for evolution by natural selection. It gives species the ability to adapt to changing conditions, new diseases, climate shifts, different predators. While it's definitely more complex, slower, and energetically costly, you need specialized organs, mating rituals, etc., the variation it creates is a powerful survival strategy in the long run.
So asexual is like photocopying, efficient but risky. Sexual is like shuffling the deck, slower but creates more possibilities. Let's look at the different ways asexual reproduction happens, starting with the really simple methods.
Certainly. One of the most straightforward is binary fission, common in unicellular organisms. Here, a single parent cell just divides into two identical daughter organisms, literally pinching itself in half. Classic examples are amoeba, paramecium. Also leishmania, the protozoan causing Kala azar, it actually divides along the specific plane, which is interesting. A variation is multiple fission. Here, a single unicellular organism produces many offspring all at once. Instead of just two, the nucleus divides repeatedly inside the parent cell, forming lots of daughter nuclei. These then get bits of cytoplasm and become new individuals before the parent cell bursts open. The malaria parasite, Plasmodium, does this inside red blood cells, allows for really rapid multiplication.
Right, overwhelming the host. And for organisms that are multicellular but still fairly simple?
For them, there's fragmentation. The parent organism literally breaks into several pieces or fragments, and then each fragment has the ability to grow and develop into a complete new organism. You see this in green algae like Spirogyra. But remarkably, also in some animals like sea anemones, starfish arms, and certain flatworms, if you cut them up, pieces can regrow.
That's amazing, like starfish regrowing arms, but the arm potentially growing a new starfish.
Sometimes, yes. Depends on the species and how much tissue is there. Then there's budding. Here, a small outgrowth, a bud, forms on the parent's body. This bud grows, develops the necessary parts, eventually detaches, and then grows into a new, independent individual. It's like a miniature version growing off the parent side. Hydra, the little freshwater polyp, and yeast, the single-celled fungus, are classic examples. Another fascinating process, though not always strictly reproduction, is regeneration. This is the ability to repair or regrow missing body parts. For some animals, if the missing part is substantial enough and contains the right stem cells, it can develop into a whole new organism. Planaria, the flatworm, is famous for this. Cut one in half, you get two worms. Hydra also has amazing regenerative powers. It shows incredible developmental potential in some organisms.
In plants, they have their own clever asexual tricks, the ones gardeners use all the time, right?
Oh, absolutely. Plants are masters of vegetative propagation. This is asexual reproduction, where new plants grow from the vegetative parts of the parent, stems, roots, or leaves, rather than from seeds. It's huge in horticulture. Think about the eyes on a potato. Those are buds. Plant a piece of potato with an eye, and you get a whole new potato plant, genetically identical to the parent.
So what are the common methods people use?
Several common techniques. Cuttings may be the simplest. You take a piece of stem or leaf, stick it in soil or water, and it develops roots and shoots. Roses, money plants, sugarcane are often done this way. Layering is where you bend a stem down to the ground, cover part of it with soil, and let it root while still attached to the parent. Once it has roots, you cut it free. Lemon, jasmine, strawberries are often layered, and grafting is more complex. You join a cutting from one plant, called the scion, onto the rooted stem of another, the stalk. They fuse and grow as one. Often used for fruit trees, like apples, citrus, or roses, to combine desirable traits, maybe tasty fruit from the scion, on disease-resistant roots from the stalk.
What are the big advantages of doing it this way?
Well, it often saves a lot of time compared to growing from seed. It allows you to propagate plants that might not produce viable seeds or produce them very slowly. And crucially, it ensures the new plants are genetically identical clones of the parent, preserving specific desirable traits like a particular flower color or fruit quality.
And then there's the really high-tech version, tissue culture. Sounds like lab work.
It absolutely is. Tissue culture, or micropropagation, is a scientific artificial form of vegetative propagation. You take tiny pieces of plant tissue, sometimes just a few cells, from a parent plant. You culture these tissues in a sterile lab environment on a special nutrient jelly, often with plant hormones added to encourage growth. These cells can multiply and form a clump called a callus, which can then be induced to form roots and shoots. eventually growing into whole new plantlets. It's incredibly efficient for mass-producing large numbers of identical plants quickly, especially for ornamentals like orchids or snake plants, or for generating disease-free planting stock. It can be costly for some plants, but it's revolutionized plant breeding. And one more asexual method, spore formation. This is common in fungi, like rhizopus, that fuzzy black bread mold, and also in non-flowering plants like ferns and mosses. Specialized structures like sporangia and fungi produce huge numbers of tiny, lightweight reproductive cells called spores. These spores are released often into the air, and if they land in a suitable environment, damp, food source, they germinate and grow into new organisms. It's a fantastic way to disperse widely.
Okay, let's switch to sexual reproduction, starting with plants. The flower is the star here, right? Nature's PR agent for reproduction.
Couldn't have said it better. The flower is absolutely the reproductive organ of the plant. It's evolved into this incredible diversity of shapes, sizes, colors, scents, all designed primarily to facilitate pollination and ensure the next generation. Flowers come in different setups. You have bisexual flowers, also called monoecious or perfect flowers. These have both male and female reproductive parts within the same flower structure. Think of a hibiscus, sunflower, rose, lily. They could potentially self-pollinate.
And the alternative.
The alternative is unisexual flowers, also called imperfect flowers. These have only male parts or only female parts. Examples include plants like papaya, cucumber, squash, watermelon. Now, on these plants, you might have separate male and female flowers on the same plant, or you might have entirely separate male plants and female plants. This necessitates cross-pollination.
So what are these male and female parts inside the flower? What do they do?
Let's break it down. The male reproductive parts are collectively called the stamen. Each stamen usually consists of a stalk called the filament, which holds up the anther. The anther is the crucial bit. It's where the pollen grains are produced. Pollen contains the plant's male gametes. The female reproductive parts are collectively called the pistil or sometimes the carpel. This typically has three parts. At the top is a stigma, which is often sticky or feathery, perfectly designed to catch pollen grains. Connecting the stigma down to the base is the style, a stalk-like structure. And at the base is the ovary, which contains one or more ovules. Inside each ovule is the female gamete, the egg cell. After fertilization, these ovules will develop into seeds.
Okay, so pollen needs to get from the anther to the stigma. That's pollination, right? How does that happen?
Exactly. Pollination is just that. The transfer of pollen from an anther to a stigma. It can happen in two main ways. Self-pollination is when pollen lands on the stigma of the same flower or another flower on the same plant. Reliable but less genetic mixing. Cross-pollination is when pollen travels from the anther of a flower on one plant to the stigma of a flower on another plant of the same species. This promotes genetic diversity.
And how does the pollen travel? It can't walk.
No, it needs help. It relies on pollination agents. Wind is a major one for many plants like grasses, oaks, ragweed. They produce tons of light pollen that just blows around anemophily. Water is used by some aquatic plants, hydrophily, but the most famous agents are animals, especially insects, entomophily, bees, butterflies, moths, flies, beetles. They visit flowers for nectar or pollen and inadvertently carry pollen from one flower to another. Birds, especially hummingbirds, and even bats can also be important pollinators. It's a beautiful coevolutionary dance.
Okay, so pollen lands on the stigma. Then what happens inside the flower to lead to a seed and maybe a juicy fruit?
Right. Once a compatible pollen grain lands on the stigma, it germinates. It grows a tiny tube, the pollen tube, down through the style, heading for the ovary. This tube carries the male gametes to an ovule. Fertilization occurs when one male gamete fuses with the egg cell inside the ovule, forming a zygote. This single-celled zygote then starts dividing and developing into an embryo, the baby plant, all still within the ovule. The ovule itself matures. It develops a protective coat and becomes the seed, containing the embryo and a food supply. Simultaneously, the ovary surrounding the ovule starts to swell and ripen, transforming into the fruit. The fruit's job is primarily to protect the seeds and often to aid in their dispersal, for example, by being eaten by animals. The other flower parts, petals, sepals, stamens, usually wither and fall off.
That's the standard path. But you mentioned earlier nature loves exceptions. Are there weird fruits out there?
Oh, absolutely. Fruit development has some fascinating twists. You can have a false fruit where the fleshy part develops not just from the ovary, but significantly from other parts of the flower, like a receptacle or thalamus. Apples and pears are classic examples. The main part we eat is swollen thalamus. Cashews, too, the apple part is a swollen stem.
So the apple isn't just the ovary, huh?
Nope. Then there are parthenocarpic fruits. These are really neat. They develop without fertilization occurring. This means they are typically seedless. Bananas are a prime example. Many varieties of grapes, cucumbers, oranges, and watermelons are also parthenocarpic, either naturally or induced by growers.
Seedless grapes. Makes sense now. Any others?
One structural type is the drupe fruit. This is defined by having a fleshy outer part surrounding a hard, stony shell, the endocarp, which encloses a single seed. Think peaches, plums, cherries, mangoes, and coconuts. A coconut is a classic drupe, though we usually see it with the fleshy outer layer removed. The hard shell is the endocarp, the white meat is the seed's endosperm, and the water is liquid endosperm.
Coconut as a drupe. Interesting. Okay, from plants, let's move to the complexities of sexual reproduction in humans. It's a very precise hormonal dance, isn't it?
It truly is. A highly coordinated sequence ensuring species continuation. Let's start with the male reproductive system. The primary organs are the testes, held outside the main body cavity in the scrotum, which helps maintain a slightly cooler temperature optimal for sperm production. Their main job is producing sperm, the male gametes, continuously, typically starting around puberty, age 10, 12 or so. From the testes, sperm mature and travel through a long duct called the vas deferens, sperm duct. Eventually, sperm exit the body via the urethra, which in males serves as a common passage for both sperm during ejaculation and urine during urination, though not at the same time. Before ejaculation, sperm mixes with fluids from accessory glands like seminal vesicles, prostate gland, to form semen. This fluid nourishes and protects the sperm. For male sterilization, a vasectomy surgically cuts or blocks the vas deferens, preventing sperm from becoming part of the semen. Now, the female reproductive system. The primary organs are the ovaries, located inside the pelvic cavity. What's truly remarkable is that a female is born with all the immature eggs or ovules she will ever possess stored in her ovaries, a finite supply. These ovules begin to mature and be released, usually one per month, starting at puberty around age 11-12. The first menstrual period is called menarche, typically around 12-13, signaling the start of fertility. The released ovule travels down the fallopian tube or oviduct towards the uterus.
And the uterus is where pregnancy develops.
Precisely. The uterus is a muscular organ where a fertilized egg implants and grows. Its inner lining, the uterine wall endometrium, thickens each month in preparation for a possible pregnancy. If no pregnancy occurs, this lining is shed during menstruation. The vagina connects the uterus to the outside. Eventually, usually in her 40s or 50s, a woman experiences menopause, the natural cessation of menstruation and reproductive capability due to declining hormone levels. It's also important to mention cervical cancer, often caused by the human papillomavirus, HPV, a common STI for which vaccines are now available. Female sterilization, tubectomy, involves surgically blocking or cutting the fallopian tubes to prevent the egg from reaching the uterus or sperm from reaching the egg.
So once the egg and sperm are potentially in the same place, what's the sequence leading to a new life?
It's a cascade of events. First, gametogenesis, making sperm and eggs. Then insemination, sperm entering the female tract. Then the crucial moment, fertilization, the fusion of one sperm and one egg, typically happening in the fallopian tube. This creates the zygote, the first single cell of the new individual. This zygote starts dividing rapidly as it travels to the uterus. Once there, it undergoes implantation, embedding itself in the prepared uterine wall. This marks the beginning of gestation, or pregnancy. The developing organism, now an embryo, continues to grow within the uterus, supported by the amazing placenta.
The placenta, that's the lifeline, right?
Absolutely. The placenta develops as an interface between mother and fetus. It facilitates the exchange of everything needed. Umbilical arteries carry deoxygenated blood and waste from the fetus to the placenta. Umbilical veins carry oxygenated blood and nutrients from the placenta to the fetus, a remarkable two-way exchange system, all without the bloodstreams directly mixing, usually.
And for those who face challenges conceiving naturally, modern medicine offers options like IVF.
Yes, IVF, or in vitro fertilization, is a major assisted reproductive technology. In vitro means in glass. Eggs are retrieved from the woman's ovaries and fertilized with sperm in a laboratory dish. If fertilization is successful, the resulting embryos are cultured for a few days, and then one or more are transferred into the woman's uterus, hopefully leading to implantation and pregnancy. It's given hope to millions.
And fertilization itself, it always happens inside the female in mammals, but not always in other animals.
Correct. We distinguish between internal fertilization, where sperm fuses with the egg inside the female's body, like in mammals, birds, reptiles, insects. This protects the gametes in developing embryo. And external fertilization, where fusion happens outside the female's body, typically in water. Think of fish releasing eggs and sperm into the water, or frogs, corals, many aquatic invertebrates like earthworms and crabs. Requires a moist environment. And then there's a really fascinating outlier in some animals. Parthenogenesis. This is essentially a form of asexual reproduction where an embryo develops from an unfertilized egg, no sperm involved. Honeybees are a classic example. Males, drones, develop from unfertilized eggs. Some lizards, fish, amphibians, and even turkeys can reproduce this way, sometimes when males are scarce. Nature's backup plan.
Wow. And finally, just a quick recap on the genetic basis and developmental stages.
Sure. Humans have 23 pairs of chromosomes in most cells, 46 total. One pair determines sex, XX for female, XY for male, developmentally. The fertilized egg is the zygote. After implantation and early cell division differentiation in the uterus, it's called an embryo. Once major body parts become distinct, around 8 weeks, it's referred to as a fetus until birth.
That's an incredibly detailed look at reproduction. Now let's shift to how organisms, especially complex ones like us, sustain themselves day to day. We need to perform certain fundamental life processes. What are these core components keeping the biological engine running?
Think of them as the absolute essentials for staying alive. The 4 crucial life processes are typically considered: digestion, getting nutrients; respiration, getting energy from nutrients; circulation, transporting substances; and excretion, getting rid of waste. These are the interconnected functions that acquire fuel, distribute it, use it, and clean up afterwards. Without any one of them, life stops.
Let's start with digestion, breaking down food for energy. It all revolves around the alimentary canal, the food highway. Can you trace the path food takes and what happens at each major stop?
Absolutely. The alimentary canal is essentially one long, muscular tube running through the body, specialized for processing food. It starts at the mouth, or buccal cavity, where food enters, then down the esophagus, food pipe via peristalsis, into the stomach, then the long, coiled small intestine, followed by the large intestine, then temporary storage in the rectum, and finally, undigested waste exits via the anus. It's crucial to remember that organs like the liver and pancreas are vital accessory digestive organs. They secrete essential juices into the canal, but food doesn't pass through them.
Okay, so the journey begins in the mouth. What happens first?
In the mouth, digestion kicks off both mechanically and chemically. Mechanical breakdown happens via chewing by the teeth, grinding food into smaller pieces, increasing surface area. Chemical digestion begins instantly with saliva, produced by salivary glands. Saliva contains the enzyme salivary amylase, also called ptyalin. This enzyme starts breaking down complex carbohydrates, like starches found in bread or potatoes, into simpler sugars like maltose. That's why starchy foods taste slightly sweet if you chew them long enough.
And then down to the stomach, the famous acid bath. What's the purpose there?
Right, the stomach is a muscular J-shaped sac. It churns the food, mixing it with acidic gastric juices. The stomach lining secretes hydrochloric acid, HCl, creating a very acidic environment, maybe pH 1.5 to 3.5. This acid does 2 main things. It kills most harmful bacteria that came in with the food, acting as a sterilizer. And it creates the perfect conditions for the stomach's main enzyme, pepsin, to work. Pepsin begins the chemical digestion of proteins, breaking them down into smaller chains called polypeptides. Sphincter muscles at the entrance and exit control how long food stays in the stomach for this churning and protein digestion before moving on.
Okay, after the stomach turns things up, food moves into the small intestine. This is often called the main site of digestion and absorption. Why is it so important?
The small intestine is absolutely the powerhouse of digestion and absorption. It's incredibly long, maybe 7 to 8 meters, coiled up tightly. It's divided into 3 sections. The duodenum, first part where most chemical digestion happens, the jejunum, and the ileum, last part where most absorption occurs. Its primary job is to finish breaking down carbohydrates, proteins, and fats into small molecules that can be absorbed into the bloodstream.
Oh.
And it's incredibly efficient at absorption because its inner lining is folded and covered in millions of tiny, finger-like projections called villi. These villi themselves have even tinier projections called microvilli. All this folding massively increases the surface area like the size of a tennis court, maximizing nutrient uptake.
Wow, a tennis court inside you. And it gets help from those accessory organs, right? The liver and pancreas.
Exactly. They deliver crucial digestive juices into the duodenum, the first part of the small intestine. The liver, our largest internal organ and gland, produces bile juice. Bile gets stored and concentrated in the gallbladder, then released into the small intestine. Bile doesn't contain digestive enzymes itself, but its key role is to emulsify fats. It breaks down large fat globules into tiny droplets, like dish soap breaking up grease. This hugely increases the surface area for fat-digesting enzymes to work on. The liver also does tons of other vital things, processing nutrients, detoxifying blood, making proteins, storing glycogen. It has specialized cells called Kupffer cells involved in immune function too. The pancreas, nestled behind the stomach, is the second largest gland and secretes powerful pancreatic juice. This juice is packed with enzymes. Pancreatic amylase continues carbohydrate breakdown. Pancreatic lipase breaks down fats, already emulsified by bile, into fatty acids and glycerol. And trypsin and other proteases continue protein breakdown into amino acids. Importantly, pancreatic juice and bile are alkaline, neutralizing the stomach acid and creating the optimal pH for these enzymes to work in the small intestine. So by the time food leaves the small intestine, digestion is largely complete, and most nutrients have been absorbed.
Okay, so carbs, fats, proteins, all broken down and absorbed in the small intestine. What's left for the large intestine?
The large intestine is shorter, maybe 1.5 meters, and wider. Its main job is to absorb water from the remaining indigestible food matter. It also absorbs some electrolytes and vitamins produced by gut bacteria living there. It compacts the leftover waste material into feces. It's composed mainly of the cecum, a pouch where the appendix attaches, and the colon, ascending, transverse, descending. Finally, the feces are stored in the rectum before being eliminated from the body through the anus during defecation. It's basically the water reclamation and waste compaction center.
Got it. Okay, let's shift to the next crucial process, respiration. The energy-releasing exchange. What's the core definition here, and how is it different from just breathing? People often use them interchangeably.
That's a really common confusion. Respiration, in the biological sense, is a fundamental biochemical process happening inside the cells of all living organisms. It's the controlled, step-by-step breakdown of organic molecules, primarily glucose, often using oxygen, to release the chemical energy stored within them. This released energy is captured in the form of ATP, adenosine triphosphate, the universal energy currency that powers almost all cellular activities. It's how cells get usable energy. Breathing, on the other hand, is simply the physical mechanical process of ventilation, taking air containing oxygen into the lungs and expelling air containing carbon dioxide out of the lungs. So breathing gets the oxygen to the cells, but respiration is what the cells do with that oxygen to make energy. Breathing is gas exchange. Respiration is energy release.
Okay, breathing is the delivery, respiration is the usage. How do plants manage this? They don't have lungs?
Right, plants have different strategies. Respiration happens in all living plant cells, day and night. They need energy too. Gas exchange occurs through various surfaces. Roots can absorb oxygen dissolved in soil water or from air spaces in the soil, diffusing through root hairs and surface cells, eplema. Leaves in young stems have tiny pores called stomata, usually more numerous on the underside of leaves. Each stoma is flanked by two guard cells that regulate its opening and closing. This controls the intake of CO2, needed for photosynthesis during the day, and the release of O2, a byproduct of photosynthesis and needed for respiration, and water vapor, transpiration. Older, woody stems have structures called lenticels for gas exchange, as the main bark becomes impermeable.
And for animals like us, what's the pathway air takes to get deep inside where the exchange happens?
In humans, the respiratory tract is a specialized pathway. Air enters through the nostrils, where it's filtered by hairs and mucus, warmed and humidified. It then passes through the pharynx, throat, a shared passage for air and food. Then into the larynx, voice box, which contains the vocal cords. You can feel it as the Adam's apple, especially in males. Below the larynx is the trachea, windpipe. This tube is kept open by C-shaped rings of cartilage, preventing collapse. At the top of the larynx, the epiglottis acts like a lid, closing off the trachea during swallowing to prevent food going down the wrong way. The trachea branches into two main bronchi, one leading to each lung. Inside the lungs, the bronchi divide repeatedly into smaller and smaller tubes called bronchioles. These bronchioles eventually end in clusters of tiny, thin-walled air sacs called alveoli. There are millions of them.
The alveoli, that's where the real action happens, the gas exchange with the blood.
Exactly. The alveoli are the functional units of the lungs for gas exchange. Each tiny alveolus is surrounded by a dense network of pulmonary capillaries, the smallest blood vessels. The walls of the alveoli and the capillaries are extremely thin, just one cell thick. This short distance allows for efficient diffusion of gases. Oxygen, which is in high concentration in the inhaled air within the alveoli, diffuses across these membranes into the blood in the capillaries, where oxygen concentration is lower. It binds to hemoglobin in red blood cells. At the same time, carbon dioxide, a waste product of cellular respiration, is in high concentration in the blood returning from the body tissues. It diffuses from the blood, across the capillary and alveolar walls into the alveoli, where its concentration is lower, ready to be exhaled. It's all driven by differences in partial pressures or concentrations.
And what's mechanically causing the air to move in and out, the diaphragm and ribs?
Precisely. Breathing mechanics involve changes in the volume of the chest cavity, driven by muscles. During inhalation, breathing in, the main muscle is the diaphragm, a large dome-shaped muscle below the lungs. It contracts and flattens, moving downward. At the same time, the external intercostal muscles between your ribs contract, pulling the rib cage upward and outward. Both actions increase the volume of the chest cavity. This causes the pressure inside the lungs to drop below atmospheric pressure, and air rushes in to equalize it, the lungs inflate. Exhalation, breathing out during quiet breathing, is mostly passive. The diaphragm relaxes and moves back up into its dome shape. The intercostal muscles relax, letting the rib cage move downward and inward. This decreases the chest cavity volume, increasing the pressure inside the lungs above atmospheric pressure, forcing air out the lungs, deflate, or recoil. It's worth noting the gas percentages change significantly. We inhale air that's about 21% O2 and 0.04% CO2, plus about 78% nitrogen. We exhale air that's about 16% O2, we don't use it all, but around 4.4% CO2, reflecting the waste product of metabolism. The nitrogen percentage stays roughly the same. And this whole rhythmic process is controlled, largely unconsciously, by the respiratory center and the medulla oblongata of the brainstem.
Okay, one last distinction in respiration. Aerobic versus anaerobic. Depends on oxygen, right?
Exactly. It's all about whether oxygen is available and used. Aerobic respiration occurs in the presence of oxygen. This is the highly efficient pathway used by most organisms, including us, most of the time. It involves the complete breakdown of glucose or other fuels into carbon dioxide and water, releasing a large amount of energy yielding about 36 to 38 ATP molecules per glucose molecule. Very efficient. Anaerobic respiration occurs in the absence of oxygen. It's a much less efficient process, yielding only a small amount of ATP, typically just 2 ATP per glucose. Because the breakdown is incomplete, it produces different end products. In human muscle cells during strenuous exercise, when oxygen supply can't keep up with demand, the end product is lactic acid. This buildup contributes to muscle fatigue and soreness. In some other organisms, like yeast, anaerobic respiration is called fermentation. Yeast fermentation produces ethanol, alcohol, and carbon dioxide. This is harnessed in baking. CO2 makes bread rise. And brewing, ethanol is the alcohol in beer and wine. And ADH is a key electron carrier molecule involved in shuttling energy during both processes, but its energy is much more effectively harvested in the aerobic pathway's electron transport chain. And remember, different animals have evolved diverse ways to get oxygen. Insects use trachea, fish use gills, amphibians can use gills, lungs, and skin. It all depends on their environment and lifestyle.
Okay, from the large-scale systems keeping us alive, let's zoom all the way down to the absolute fundamental level. The cell. It's always called the basic structural and functional unit of life. What does that really mean in practice?
It means the cell is the smallest thing we can point to and say, that's alive. It's the minimum package that can perform all the essential functions of life. It can grow, it can metabolize, process energy, it can respond to stimuli from its environment, it can reproduce itself, and it can maintain a stable internal environment, homeostasis. Just like a single brick is the basic unit of a wall, capable of bearing load and having structure, the cell is the fundamental building block of every single living organism, from bacteria to blue whales. Every complex organism starts as just one cell and then builds upon that, with different cells specializing to perform all the necessary tasks for the whole organism to survive.
And discovering this microscopic world must have been revolutionary. Who are the key players that opened up this field of cell biology?
Oh, absolutely revolutionary. It really hinged on the invention and improvement of the microscope. The story usually starts with Robert Hooke in 1665. He looked at thin slices of cork under his early microscope and saw these tiny, empty boxes. They reminded him of monks' cells in a monastery, so he coined the term cells. He was seeing dead plant cell walls, but it was the first glimpse. He's often called the father of cytology.
So Hooke saw the boxes. Who saw things inside the boxes?
That was Antony van Leeuwenhoek, around 1674. He was a Dutch cloth merchant, but an amazing lens grinder. His microscopes were much more powerful. He looked at pond water, teeth scrapings, all sorts of things, and saw living, moving, microscopic organisms, bacteria, protozoa, which he called animalcules. He was the first to see living cells in action.
Incredible. And then in the 1800s, things really took off.
Yes, the 19th century was huge for cell biology. In 1831, Robert Brown identified the nucleus as a distinct structure within plant cells. Then came the foundational cell theory. primarily from Matthias Schleiden, a botanist, 1838, and Theodor Schwann, a zoologist, 1839. They proposed that all plants, Schleiden, and animals, Schwann, are composed of cells, this unified biology. Around the same time, Purkinje in 1839 coined the term protoplasm for the living substance inside the cell.
What about where new cells come from? Did they think they just popped into existence?
That was debated. But Rudolf Virchow in 1858 famously stated, omnis cellula e cellula. All cells arise from pre-existing cells. This firmly established cell division is the basis of reproduction and growth, refuting spontaneous generation for cells. Other key discoveries followed. Friedrich Mischer discovered DNA back in 1869, though its importance wasn't immediately clear. Altmann discovered mitochondria, 1886. Camillo Golgi discovered the Golgi apparatus, 1898. Walter Fleming observed chromosomes during cell division and coined chromatin, 1882, and then the massive breakthrough in 1953. Watson and Crick, using critical X-ray diffraction data from Rosalind Franklin and Maurice Wilkins, figured out the double helix structure of DNA. This unlocked the secrets of heredity at the molecular level. These pioneers truly built our modern understanding of the cell.
Amazing history. Okay, let's take a tour inside a typical cell. What are the main components, the organelles, and what jobs do they do? Starting from the outside in?
All right, so on the very outside of many cells plants, fungi, bacteria, but not animals, you have the cell wall. This is a rigid outer layer that provides structural support, protection, and determines the cell's shape. Its composition varies, peptidoglycan in bacteria, cellulose in plants, chitin in fungi. Animal cells lack this, allowing them more flexibility. Just inside the cell wall, or as the outer boundary in animal cells, is the cell membrane, or plasma membrane. This is a crucial, selectively permeable barrier. Think of it as the cell's bouncer. It controls what gets in and out. It's made primarily of a double layer of phospholipids with various proteins embedded in it, allowing for transport, signaling, and adhesion.
Okay, so wall for structure, if present, membrane for control. What's the filling inside?
The jelly-like substance filling the cell, suspending all the organelles, is the cytoplasm. It's mostly water, but packed with salts, proteins, and other molecules. It's where many metabolic reactions happen. Now, for the control center, especially in eukaryotic cells, the nucleus. It's usually the largest organelle enclosed by a double membrane, the nuclear envelope. Its main job is to house the cell's genetic material DNA. Inside the nucleus, you find the nucleolus, which is where ribosomes are made. The DNA itself isn't just floating around, it's organized into chromatin DNA wound around proteins, histones. During cell division, chromatin condenses to form visible chromosomes. Humans have 23 pairs.
The nucleus holds the blueprints. What about the power plant?
That would be the mitochondria, singular, mitochondrion, famously known as the powerhouse of the cell. These are where cellular respiration occurs, breaking down glucose to produce the vast majority of the cell's energy currency, ATP. They have two membranes, a smooth outer one and a highly folded inner membrane. Those folds are called cristae, and they massively increase the surface area for ATP production. What's super interesting is that mitochondria have their own circular DNA and ribosomes, suggesting they evolved from ancient bacteria that were engulfed by early eukaryotic cells. ATP, remember, fuels almost everything the cell does.
Okay, powerhouse covered. What about manufacturing and transport within the cell, like a factory and delivery service?
Good analogy. That involves the endoplasmic reticulum, ER, and the Golgi apparatus. The ER is a vast network of interconnected membranes spreading throughout the cytoplasm. There are two types. Rough ER looks rough because it's studded with ribosomes. Its main job is synthesizing and modifying proteins, especially those destined to be secreted from the cell or embedded in membranes. Smooth ER lacks ribosomes, looks smoother and more tubular. It's involved in synthesizing lipids, fats and steroids, metabolizing carbohydrates, and crucially, detoxifying drugs and poisons. Liver cells, which do a lot of detox, are packed with smooth ER. Proteins and lipids made in the ER often then move to the Golgi apparatus, or Golgi complex body. Think of the Golgi as the cell's post office, or finishing and shipping center. It receives proteins and lipids from the ER, further modifies them, sorts them, and packages them into vesicles, little membrane bubbles, for delivery to their final destinations either elsewhere in the cell or secreted outside. It's made of flattened sacs called cisternae.
And the ribosomes themselves, you mentioned them on the rough ER.
Right. Ribosomes are the actual protein synthesis machines. They're tiny structures made of ribosomal RNA, rRNA, and protein. They read the instructions carried by messenger RNA, mRNA, from the DNA, and link amino acids together in the correct sequence to build a specific protein. They can be found free in the cytoplasm or attached to the rough ER. Other important organelles include vacuoles. These are membrane-bound sacs used for storage water, nutrients, ions, waste products. Plant cells typically have a very large central vacuole that helps maintain turgor pressure. Animal cells have smaller, more numerous vacuoles, if any. Peroxisomes are small organelles involved in metabolic processes that produce hydrogen peroxide, which they then safely break down. They help detoxify substances and break down fatty acids. And lysosomes, the suicide bags or recycling centers. Formed by the Golgi, they contain powerful digestive enzymes made initially on the rough ER. They break down worn-out organelles, cellular debris, ingested bacteria, etc. If a cell is severely damaged, lysosomes can rupture and digest the cell itself.
And plant cells have some extras, right? Especially for making food.
Yes. Plant cells have plastids. These are major organelles, often involved in photosynthesis or storage. Like mitochondria, they have their own DNA and ribosomes. The most famous are chloroplasts. These contain the green pigment chlorophyll and are the site of photosynthesis. Inside, you have stacks of disc-like structures called grana, singular.granum, made of individual thylakoids where the light-dependent reactions happen. The fluid-filled space around them, the stroma, is where the light-independent Calvin cycle reactions occur, fixing CO2 into sugar. They are the plant's solar panels and sugar factories. Other plastids include chromoplasts, which synthesize and store pigments other than chlorophyll: yellows, oranges, reds, like carotene in carrots, lycopene in tomatoes, betaine in beets. They give fruits and flower their colors. And leucoplasts, which are non-pigmented and primarily used for storage. Amyloplasts store starch, elaioplasts store fat soils, and aleuroplasts store proteins. Very important for energy reserves in roots, seeds, etc.
So that's the toolkit inside. How do we classify the cells themselves on a broader scale? Prokaryotic versus eukaryotic.
That's the most fundamental division of cell types. Prokaryotic cells, pro means before, karyon means nucleus, are simpler, generally smaller, and evolutionarily older. Their defining features are the lack of a true membrane-bound nucleus and the absence of most membrane-bound organelles, like mitochondria, Golgi. Their genetic material, usually a single circular DNA molecule, floats in the cytoplasm in a region called the nucleoid. Bacteria and archaea are prokaryotes. Eukaryotic cells, this is true, are more complex, generally larger, and evolved later. They have a well-defined membrane-bound nucleus containing linear DNA organized into chromosomes. They also possess a variety of membrane-bound organelles, allowing for compartmentalization and specialization of functions. Plants, animals, fungi, and protists are all eukaryotes. This complexity allowed for the evolution of multicellular life.
Okay, and within eukaryotes, what are the key differences between a typical plant cell and an animal cell again?
Sure. While both are eukaryotic, they have distinct features reflecting their lifestyles. Plant cells typically have 1. A rigid cell wall made of cellulose outside the cell membrane for support and protection. 2. Chloroplasts for photosynthesis. 3. A large central vacuole for storage and maintaining turgor pressure. They often have a more fixed, somewhat rectangular shape. Animal cells, on the other hand, 1. Lack a cell wall. 2. Lack chloroplasts. 3. Usually have small vacuoles, if any. They tend to have more irregular, rounded shapes. A feature often present in animal cells and lower plants, but absent in higher plants, are centrioles, which are involved in organizing microtubules during cell division.
That brings us perfectly to how cells replicate. Cell division, essential for growth, repair, making new organisms. Can you walk us through the cell cycle and the main types of division?
The life of a dividing cell follows a regulated sequence called the cell cycle. It has two main phases: interphase, the growth and preparation phase, and the M phase, the actual division phase. Interphase is the longest part. It's not a resting phase. The cell is very active metabolically. It's subdivided into G1 phase, gap 1. Cell grows, makes proteins and organelles, carries out normal functions. S phase, synthesis. This is crucial. The cell replicates its entire DNA. Each chromosome is duplicated. G2 phase, gap 2. Cell continues to grow, makes proteins needed for division, checks the duplicated DNA for errors, and prepares for mitosis. Some cells, once mature, exit the cycle and enter a non-dividing state called G-row phase. Nerve cells, muscle cells often do this. Following interphase is the M phase, which includes nuclear division and cytoplasmic division. There are two main types of nuclear division: mitosis. This involves one round of division and produces two daughter cells that are genetically identical to the parent cell and have the same number of chromosomes, diploid in humans. Mitosis is used for growth, tissue repair, and asexual reproduction. It has distinct stages: prophase, metaphase, anaphase, telophase, followed by cytokinesis, division of the cytoplasm, meiosis. This is a more specialized type of division used for sexual reproduction. It involves two rounds of division, meiosis the first and meiosis the second, and produces four daughter cells, gametes like sperm and eggs, that are genetically non-identical to the parent cell and have half the number of chromosomes, haploid. This reduction in chromosome number is essential, so that when sperm and egg fuse, the correct diploid number is restored. Meiosis introduces genetic variation through processes like crossing over. Each meiotic division also has prophase, metaphase, anaphase, and telophase stages.
And that DNA replication in the S phase, copying the entire genetic code, sounds incredibly complex. What are the key molecular players involved, like enzymes?
It is an amazing molecular ballet. DNA replication is semi-conservative. Each new DNA molecule has one old strand and one new strand, and incredibly accurate. It relies on a whole team of enzymes and proteins. Helicase, the unzipper. It unwinds the DNA double helix by breaking the hydrogen bonds between the base pairs. Single-strand binding proteins, SSBs. These bind to the separated strands to keep them from snapping back together. Topoisomerase relieves the twisting stress that builds up ahead of the replication fork as the DNA unwinds. Primus synthesizes short RNA primers, which provide a necessary starting point for the DNA polymerase. DNA polymerase can't start a new strand from scratch. DNA polymerase III, the main builder. It reads the template strand and adds complementary DNA nucleotides to synthesize the new strand in the 5' direction. DNA polymerase I removes the RNA primers and replaces them with DNA nucleotides. DNA leaves it. The gluer. It joins the Okazaki fragments, short pieces synthesized on the lagging strand, and seals any nicks in the DNA backbone, creating continuous strands. Telomerase. In eukaryotes, this enzyme helps maintain the ends of linear chromosomes, telomeres, preventing them from getting shorter with each replication. It's a highly coordinated, high-fidelity process essential for life.
Wow. Okay, one last thing on cells. How do things, especially water, move across that cell membrane? Diffusion and osmosis.
Yes, those are two key passive transport mechanisms. Diffusion is the general tendency for molecules to spread out from an area of higher concentration to an area of lower concentration, just due to their random motion. Think of the smell of coffee spreading through a room. It happens down a concentration gradient and doesn't require the cell to expend energy. Many small nonpolar molecules can diffuse across the cell membrane. Osmosis is a specific type of diffusion. It's the diffusion of water across a selectively permeable membrane, like the cell membrane. Water moves from an area of low solute concentration, which means high water concentration, to an area of high solute concentration, low water concentrations. Basically, water moving to dilute the more concentrated solution, trying to equalize concentrations on both sides.
And this affects cells placed in different solutions, right? Hypotonic, hypertonic.
Exactly. The tonicity of the surrounding solution dictates water movement. If a cell is in a hypotonic solution, lower solute concentration outside than inside, water will rush into the cell via osmosis. An animal cell might swell and burst. A plant cell will swell, but the cell wall prevents bursting. It becomes turgid. If a cell is in a hypertonic solution, higher solute concentration outside, water will move out of the cell. The cell will shrink or shrivel. Crenation in animal cells, plasmolysis in plant cells. If a cell's in an isotonic solution, same solute concentration inside and out, there's no net movement of water, and the cell maintains its normal shape and volume. Understanding osmosis is crucial for everything from IV fluids to plant wilting.
Okay, from the cell as the basic unit, let's zoom out one level to tissues. What exactly defines a tissue, and why is this grouping of cells important?
Tissues are essentially groups of cells that are structurally similar and work together cooperatively to perform a specific function. This is really the first level of organization where we see a significant division of labor in multicellular organisms. Instead of every cell trying to do everything, cells start to specialize and team up. This makes the whole organism much more efficient and capable of complex tasks that a single cell just couldn't manage. Think of it like moving from individual artisans to an organized factory assembly line.
Makes sense. Let's start with plant tissues. What are the main kinds, and how do they help a plant grow and function?
Plant tissues are broadly divided into two main categories based on their ability to divide: meristematic tissue and permanent tissue. Meristematic tissues are the growth zones of the plant. Their cells are actively dividing by mitosis. They are typically small, cube-shaped, have dense cytoplasm, large nuclei, small vacuoles, and crucially, they lack intercellular spaces. They're tightly packed. They are responsible for all plant growth.
Where do you find these growth tissues?
There are three main types based on location. Apical meristem, found at the very tips of roots and shoots, stems and branches. Think of them as the primary growth points, responsible for increasing the plant's length, making roots grow deeper and shoots grow taller. Intercalary meristem, found at the base of leaves or internodes, the sections of stem between nodes or leaves, especially in grasses. It helps in the growth and elongation of these parts, allowing grasses to regrow quickly after being grazed, for example. Lateral meristem, found along the sides of stems and roots running parallel to the long axis. This is responsible for secondary growth, meaning increasing the thickness or diameter of the plant. Examples include the vascular cambium, produces new xylem and phloem, and cork cambium, produces bark. This is what makes tree trunks get wider over time.
Okay, so meristems are for growth. What happens when those cells stop dividing and take on a specific job?
That's when they become permanent tissues. These cells are derived from meristematic tissues but have lost the ability to divide actively, or divide only rarely. They have differentiated and become specialized in structure and function. Permanent tissues can be simple or complex. Simple permanent tissues are made up of only one type of cell, all working together. The most common is parenchyma. These are typically living cells with thin walls and often large vacuoles. They form the bulk of the plant body and are involved in functions like photosynthesis, if they contain chloroplasts, then called chlorenchyma, food storage, starch, oils, proteins, and secretion. Sometimes they have large air spaces, aerenchyma, providing buoyancy in aquatic plants. Then there's cholenchyma. These are also living cells, but they have unevenly thickened primary walls, especially at the corners. They provide mechanical support and flexibility to growing stems, leaf stalks, petioles, and other soft plant parts, allowing them to bend without breaking. Think of the strings in celery. In sclerenchyma, these cells have very thick, rigid secondary walls, often lignified, containing lignin, and they are usually dead at maturity. Their sole function is to provide strength, rigidity, and mechanical support to the plant. Examples include fibers, like in hemp or flax, and scleriates, the gritty cells in pear fruit or the hard shells of nuts, like the plant's internal skeleton.
Okay, those are the simple ones. What about complex permanent tissues?
Complex permanent tissues are made up of more than one type of cell, all working together to perform a common function, primarily transport. The two main types are xylem and phloem, which together form the vascular tissues of the plant. The xylem's main job is the bulk transport of water and dissolved minerals from the roots upwards to the rest of the plant. It also provides mechanical support. It consists of several cell types, tracheids and vessel elements, the main water-conducting cells dead at maturity, xylem parenchyma, living cells for storage, and xylem fibers for support. It's a one-way street for water. Phloem's main job is the transport of food sugars produced during photosynthesis, mainly sucrose, from the leaves, where they're made, to other parts of the plant where they are needed for growth or storage, like roots, fruit, seeds. This transport is called translocation and can be multidirectional, up or down. Phloem consists of sieve tubes or sieve cells, conducting cells living but lack nucleus at maturity, companion cells closely associated with sieve tubes controlling their function, phloem parenchyma for storage, and phloem fibers for support, usually the only dead cells in phloem. Together, xylem and phloem are the plant's plumbing system.
Fascinating how plants build themselves. Now, let's switch to animal tissues. They seem much more diverse in function, related to movement and complexity.
They are indeed, reflecting the active lifestyle and complex organ systems of animals. We usually classify animal tissues into four main types: epithelial, connective, muscular, and nervous tissue. Epithelial tissue, or epithelium, forms covering and lining layers. It covers the external body surface, like your skin's epidermis, lines internal organs and cavities, like the lining of your digestive tract or respiratory tract, and forms glands. Its main functions are protection, secretion, absorption, excretion, filtration, and sensory reception.
And it comes in different shapes.
Yes, epithelial tissues are classified based on cell shape and the number of layers. Shape: squamous, flat tile-like cells, good for diffusion, found in lung alveoli, lining blood vessels. Cuboidal, cube-shaped, good for secretion absorption, found in kidney tubules, glands. Columnar, tall, column-shaped, also for secretion absorption, lining stomach and intestines. Layers: simple, single layer, or stratified, multiple layers. Special types include ciliated epithelium, has cilia to move substances, lining respiratory tract, fallopian tubes. Transitional epithelium can stretch, lining urinary bladder, and glandular epithelium, specialized for secretion, forming sweat glands, salivary glands, endocrine glands. The second major type is connective tissue. This is the most abundant and widely distributed tissue type. As the name suggests, its main functions are to bind, support, protect, insulate, and transport substances. Connective tissues are diverse but share a common structure. Specialized cells scattered within an extracellular matrix. This matrix consists of protein fibers like collagen, elastin, and a ground substance, fluid, gel, or solid. Major types include dense connective tissue, packed with collagen fibers, providing strength, found in tendons, connecting muscle to bone, and ligaments, connecting bone to bone.
Tendons and ligaments, right. What about looser connective tissues?
Yes, loose connective tissue has fewer fibers and more ground substance. Areolar tissue is a common type, acting as packing material between organs, binding skin to underlying muscles. It's involved in inflammation and tissue repair. Adipose tissue is fat tissue. Its cells, adipocytes, store triglycerides. It provides insulation, cushioning for organs, and energy storage, found beneath the skin and around organs. We also have specialized connective tissues like cartilage, which is strong yet flexible, found in joints, reducing friction, ear, nose, trachea, provides support and cushioning. Bone, osseous tissue, is a hard connective tissue formed in the skeleton, provides support, protection, muscle attachment, stores calcium, and houses bone marrow, where blood cells are made. And blood and lymph are considered fluid connective tissues. Blood transports oxygen, nutrients, waste, hormones, and contains cells for immunity, all within a fluid matrix, plasma. Lymph circulates immune cells and returns fluid to the bloodstream.
Okay, covering, connecting. What about movement?
That's the job of muscular tissue. It's highly specialized for contraction, generating force to produce movement. There are 3 types. Skeletal muscle, attached to bones. Its cells are long, cylindrical, multinucleated, and have obvious striations, stripes. This is voluntary muscle. We consciously control its contraction to move our body. Smooth muscle, found in the walls of internal organs, stomach, intestines, bladder, blood vessels. Its cells are spindle-shaped, have a single nucleus, and lack striations. This is involuntary muscle. Its contractions are not under conscious control. It controls things like digestion, blood pressure regulation. Cardiac muscle, found only in the wall of the heart. Its cells are branched, usually have a single nucleus, and are striated. Like smooth muscle, it's involuntary. A unique feature is intercalated discs, specialized junctions connecting the cells that allow rapid electrical signal transmission, ensuring the heart beats as a coordinated unit.
And the 4th type, the communication network.
That's nervous tissue. It forms the brain, spinal cord, and nerves. It's specialized for detecting stimuli, processing information, and transmitting electrical signals, nerve impulses, rapidly throughout the body, coordinating body activities. The main functional cell is the neuron, nerve cell. It has a unique structure for communication. Dendrites, branched extensions that receive signals from other neurons or sensory receptors and transmit them towards the cell body, or soma. Contains the nucleus and other organelles, integrates incoming signals. Axon, a single long projection that transmits nerve impulses away from the cell body towards other neurons, muscles, or glands. Can be very long. Many axons are covered by a myelin sheath, a fatty insulating layer that speeds up impulse transmission. The synapse, the junction where a neuron communicates with the next cell, neuron, muscle, or gland, usually involves the release of chemical messengers called neurotransmitters across a tiny gap. Nervous tissue also contains supporting cells called neuroglia or glial cells.
Okay, we've built the body with cells and tissues. Now, how is it all controlled and coordinated? There are 2 main systems for that, right? The fast-acting nervous system and the slower endocrine system. Let's start with the human nervous system, the body's high-speed internet.
Exactly. The nervous system is our rapid communication network, using electrical and chemical signals. It's incredibly complex. We divide it into 2 main parts, the central nervous system, CNS, which is the command center of the brain and spinal cord, and the peripheral nervous system, PNS, which includes all the nerves that branch out from the CNS, connecting it to the rest of the body, limbs, organs, skin. It's the information highway.
The brain is clearly the main processor. Such a vital organ must be well protected, right? And what are its major functional regions?
Absolutely. The brain is incredibly well protected, first by the bony skull, cranium. Then, wrapped around the brain and spinal cord are 3 protective membranes called the meninges. Between 2 of these layers flows cerebrospinal fluid, CSF, which acts like a liquid cushion, absorbing shocks and providing buoyancy. The brain literally floats in this fluid. An adult brain weighs roughly 1.3 to 1.4 kilograms. Functionally, the brain is usually divided into 3 main parts: forebrain, midbrain, and hindbrain. The forebrain is the largest and most complex part, responsible for higher cognitive functions. It includes the cerebrum. This is the thinking part. It's responsible for voluntary actions, consciousness, learning, memory, language, personality. It's divided into 2 cerebral hemispheres, left and right, and has a highly folded outer layer, the cerebral cortex. Specific areas are lobes, specialized: frontal lobe, planning, decisions, voluntary movement; parietal lobe, processing sensory info like touch, temperature, pain, pressure, taste; temporal lobe, hearing, memory, language comprehension; and occipital lobe, vision. Note, the original outline incorrectly listed olfactory lobe here. Olfactory bulbs process smell, but visual reception is occipital. Also in the forebrain are the thalamus, which acts as a relay station, sorting and directing sensory information, except smell, to the cerebrum.
and the hypothalamus, located below the thalamus. It's small but vital, regulating body temperature, hunger, thirst, sleep-wake cycles, controlling the pituitary gland and influencing emotions.
Okay, that's the complex forebrain. What about the midbrain and hindbrain?
The midbrain is a smaller section connecting the forebrain and hindbrain. It's involved in processing visual and auditory information, controlling eye movements, and regulating reflex movements of the head and eyes in response to sights and sounds. It also controls things like pupil size. The hindbrain sits at the base of the brain, connecting to the spinal cord. It includes the cerebellum, little brain, located at the back, crucial for coordinating voluntary movements, maintaining posture, balance, and ensuring movements are smooth and precise. Doesn't initiate movement, but refines it. The pons acts as a bridge, relaying signals between the cerebrum and cerebellum and between the brain and spinal cord. Involved in regulating breathing, the medulla oblongata connects directly to the spinal cord. This is absolutely vital, controlling essential involuntary functions like heartbeat, breathing rate and depth, blood pressure, swallowing, vomiting, coughing. Damage here is often fatal.
And sometimes we react incredibly fast without thinking, like pulling your hand off a hot stove. How does that work so quickly?
Well, yeah, it's a reflex, right? Exactly. That's a reflex action. It's an involuntary, automatic, and incredibly rapid response to a stimulus. It's a protective mechanism that bypasses conscious processing in the brain for speed. The pathway is called a reflex arc. 1. Stimulus, for example, heat, activates a sensory receptor, for example, in the skin. 2. The receptor sends a signal along a sensory neuron to the spinal cord. 3. Within the spinal cord, the sensory neuron usually connects directly or via one or more inner neurons to a motor neuron. 4. The motor neuron sends a signal out to an effector, usually muscle or gland. 5. The effector produces the response, for example, muscle contracts to pull hand away. Because the processing happens directly in the spinal cord, the response occurs much faster than if the signal had to travel all the way up to the brain and back down. You often feel the pain after you've already moved your hand.
Brilliant efficiency. Now let's talk about the other control system, the endocrine system. This uses chemical messengers, hormones, right? Slower, but with widespread effects.
Precisely. The endocrine system consists of several ductless glands scattered throughout the body. These glands synthesize and secrete chemical messengers called hormones directly into the bloodstream. Hormones travel via the blood to target cells or organs, which have specific receptors for that hormone. Binding to the receptor triggers a specific response in the target cell. This system regulates slower, longer-term processes like growth, development, metabolism, reproduction, mood, and stress response.
What are some of the key endocrine glands and the hormones they produce?
Let's hit the highlights. The pituitary gland, located at the base of the brain, is often called the master gland because it produces hormones that regulate many other endocrine glands and body functions. It secretes growth hormone, somatotropin. Too little causes dwarfism, too much causes gigantism in children, or acromegaly in adults. It also releases hormones controlling the thyroid, adrenals, and gonads, plus others like ADH, water balance, and oxytocin. The thyroid gland, in the neck, produces thyroxine and T3, which regulates the body's overall metabolic rate, affecting energy levels, temperature, and development. Iodine is essential for making these hormones. Deficiency can cause goiter, enlarged thyroid, or hypothyroidism. The pancreas has endocrine functions too. Clusters of cells called islets of Langerhans produce insulin, lowers blood glucose by helping cells take it up, and glucagon raises blood glucose by stimulating the liver to release stored glucose. Essential for blood sugar regulation, problems lead to diabetes. The adrenal glands, sitting on top of the kidneys, produce several hormones. The inner part, medulla, produces adrenaline, epinephrine, and noradrenaline, responsible for the fight or flight response to stress. The outer part, cortex, produces corticosteroids like cortisol, stress response metabolism, and aldosterone, saltwater balance. Deficiency, e.g. Addison's disease, can be serious. The gonads' ovaries in females produce estrogen and progesterone, regulate female reproductive cycle, secondary sexual characteristics. Testes in males produce testosterone, regulates male development, sperm production, secondary sexual characteristics. These are the primary sex hormones.
So animals have nerves and hormones. How do plants coordinate things? They don't have nerves. Is it all hormones for them?
Pretty much, yes. Plants rely on chemical messengers called plant hormones or phytohormones to coordinate their growth, development, and responses to the environment. They're produced in one part and travel to another to cause an effect, often at very low concentrations.
What are some of the main plant hormones?
There are 5 major classes. Auxins, like IAA, primarily promote cell elongation, especially in stems and roots, involved in phototropism, bending towards light, root formation, fruit development. Produced mainly in shoot tips, gibberellins, chiaes. Also promote stem elongation, stimulate seed germination, flowering, and fruit development. Overcoming dormancy, cytokinins. Primarily promote cell division, cytokinesis, and differentiation. Involved in delaying leaf aging, stimulating branching, found in growing areas like root tips, embryos, abscisic acid, ABA, generally acts as an inhibitor. Promotes seed and bud dormancy, helps plants respond to stress, like drought by closing stomata, inhibits growth, often called the stress hormone, ethylene. Unique because it's a gaseous hormone. Promotes fruit ripening, leaf abscission, falling off, and plant aging, senescence. One bad apple spoils the bunch because the ripening apple releases ethylene gas.
And plants move in response to stimuli too, but it's usually growth, right? Tropisms.
Yes, tropism is directional growth in response to a directional stimulus. Phototropism. Growth towards positive, like shoots, or away from negative, like roots, light, driven by auxins. Geotropism, or gravitropism. Growth in response to gravity. Roots grow down, positive. Shoots grow up, negative. Also involves auxins and other factors. Hydrotropism. Growth towards water, important for roots seeking moisture. Chemotropism. Grows toward a chemical stimulus. The growth of the pollen tube towards the ovule, guided by chemicals secreted by the ovule, is a key example. Thigmotropism. Growth in response to touch, like tendrils coiling around a support.
But some plant movements aren't growth, right? Like the touch-me-not plant closing its leaves.
Right, that's nastic movement. These are non-directional responses to stimuli, usually temporary and often involving changes in turgor pressure, not growth. The direction of the response is independent of the direction of the stimulus. The rapid folding of the touch-me-not, Mimosa pudica, leaves when touched is a classic example, thigmonesty. Venus flytrap closing on an insect is another. These are rapid, often protective or predatory movements.
Okay, we've covered structure, control, reproduction. Now for the fuel itself. Nutrition. How organisms get the energy and building blocks they need. And there's some pretty diverse strategies out there, right? Not just eating like us.
Absolutely. Life has found incredible ways to get nutrients. One major strategy is saprophytic nutrition. These organisms, called saprophytes or decomposers, feed on dead and decaying organic matter, fallen leaves, dead animals, waste products. Fungi, like mushrooms, molds, and many bacteria are classic saprophytes. They secrete enzymes to digest the material externally and then absorb the nutrients. They play a vital role in recycling nutrients in ecosystems, nature's recyclers. Then there's parasitic nutrition. Here, the organism, the parasite, lives on or inside another living organism, the host, and obtains its nutrients directly from the host, often causing harm in the process. Think tapeworms in intestines, fleas on a dog, mistletoe on a tree. The parasite benefits, the host suffers. And a third strategy is symbiotic nutrition. This involves two different organisms living together in a close relationship where at least one, and often both, benefit nutritionally or otherwise. A great example is lichens, which are a partnership between a fungus and an alga or cyanobacterium. The alga photosynthesizes, providing food for both, while the fungus provides structure, protection, and absorbs water minerals. They can live in harsh places. Neither could survive alone. Another example is nitrogen-fixing bacteria in the roots of legume plants.
Fascinating strategies. Focusing on animals, especially humans, we always hear about the macronutrients. The big three, what are they and why are they so crucial?
The big three macronutrients we need in large amounts are carbohydrates, fats, lipids, and proteins. Carbohydrates are our body's primary source of energy. They're the easiest fuel for cells to use. They range from simple sugars, monosaccharides like glucose, fructose, disaccharides like sucrose, lactose, to complex carbs. Polysaccharides like starch in plants, glycogen in animals, cellulose fiber in plants. The Benedict's test is used in labs to check for simple reducing sugars. Plants synthesize glucose during photosynthesis, which is the fundamental unit. When our muscles work hard without enough oxygen, they break down glucose anaerobically, producing lactic acid, causing fatigue. Fats, lipids, are our most concentrated energy source, providing more than double the energy per gram compared to carbs or proteins. They're essential for long-term energy storage in adipose tissue, insulation, cushioning organs, absorbing fat-soluble vitamins, and building cell membranes. Types include saturated fats, usually solid at room temp, mostly from animal sources like butter, meat fat. Unsaturated fats, usually liquid from plant sources like olive oil, nuts, seeds, often considered healthier. And trans fats, often artificially created, found in some processed food like margarine, generally unhealthy. Cholesterol is a specific type of lipid needed for membranes and hormone synthesis, but high levels in blood can be problematic. And proteins, often called the bodybuilding nutrients. They are complex molecules made of long chains of amino acids, linked by peptide bonds. There are 20 common amino acids. Our bodies can make some,
But others are essential amino acids. We must get them from our diet. Okay, leucine, lysine, tryptophan, histidine. Proteins have countless functions. Building and repairing tissues, muscle, skin, hair, acting as enzymes, catalyzing reactions, forming many hormones like insulin, playing key roles in the immune response, antibodies, transporting substances like hemoglobin carrying oxygen. Animal proteins, meat, fish, eggs, dairy, are often called first-class or complete proteins because they contain all essential amino acids. Casein is the main protein in milk.
Okay, those are the macros we need lots of. But we also need tiny amounts of micronutrients, vitamins and minerals. They might be small in quantity, but they're vital, right?
Absolutely critical. Vitamins are organic compounds needed in small amounts for various metabolic processes. They were discovered by F.G. Hawkins, and the name vitamin was coined by Casimir Funk. We categorize them as water-soluble or fat-soluble. Water-soluble vitamins, B vitamins, and vitamin C are generally not stored well in the body, so we need regular intake. Let's quickly go through them. B1, thiamine. Energy metabolism, nerve function deficiency, beriberi. B2, riboflavin. Energy production deficiency, araboflavinosis, cracked lips, sore throat. B3, niacin. DNA repair, metabolism deficiency, pellagra, dermatitis, diarrhea, dementia, the 3Ds. B5, pantothenic acid, fatty acid metabolism deficiency, burning feet syndrome. B6, pyridoxine, amino acid metabolism, red blood cell formation deficiency, microcytic anemia, nerve issues. B7, biotin, metabolism deficiency, hair loss, skin rashes. B9, folate acid, DNA synthesis, cell division, crucial in pregnancy deficiency, megaloblastic anemia, neural tube defects. B12, cobalamin, nerve function, red blood cell formation, contains cobalt, deficiency, pernicious anemia, nerve damage. Vitamin C, ascorbic acid, antioxidant, collagen synthesis, immune function, deficiency, scurvy, bleeding gums, pull wound healing. Fat-soluble vitamins, A, D, E, K, can be stored in the body's fat tissues and liver, so excessive intake can be toxic. Vitamin A, retinol, vision, especially night vision, immune function, skin health. Deficiency, night blindness, dry eyes, xerophthalmia. Vitamin D, calciferol, calcium absorption, bone health, synthesized in skin with sunlight. Deficiency, rickets, children, osteomalacia, adult soft bones. Vitamin E, tocopherol, antioxidant protects cell membranes. Deficiency, rare, can cause neurological problems. Vitamin K, fuliquinone, essential for blood clotting. Deficiency, excessive bleeding.
Wow, quite the list. And what about minerals? Inorganic, but equally important.
Yes, minerals are essential inorganic elements. Calcium, chalate, most abundant mineral, crucial for bones and teeth formation and strength, also muscle contraction, nerve function, blood clotting. Deficiency leads to osteoporosis. Iron, Fe, essential component of hemoglobin and red blood cells, which carries oxygen. Deficiency is the most common cause of anemia worldwide, fatigue, weakness. Magnesium, Mg, involved in hundreds of enzyme reactions, muscle and nerve function, energy production, blood sugar control, bone health. Others like potassium, sodium, zinc, iodine, selenium are also vital in smaller amounts for various functions like fluid balance, thyroid function, immune response, etc. Micronutrients pack a punch.
Okay, that covers animal nutrition basics. How do plants get their fuel? It all comes down to that powerhouse process, photosynthesis, right?
Absolutely. Plants are autotrophs. They make their own food using photosynthesis. It's the process where green plants, algae, and some bacteria use light energy to convert carbon dioxide and water into glucose, sugar, their food, and oxygen, released as a byproduct. It underpins almost all ecosystems.
What are the essential ingredients for photosynthesis?
Plants need several key things. 1, light energy, usually from the sun. 2, water, H2O, absorbed by the roots. 3, carbon dioxide, CO2, taken from the atmosphere through stomata. 4, chlorophyll, the green pigment located in chloroplasts which absorb light energy. 5, a suitable temperature range. The overall summary equation is 6 CO2 plus 6 H2O plus light energy, C6H12O6, glucose plus 6O2.
And you mentioned stomata and guard cells before. They regulate gas exchange.
Yes. Stomata are the tiny pores, mostly on leaf undersides, that allow CO2 to enter and O2 and water vapor to exit. Each stoma is surrounded by 2 guard cells that control its opening and closing based on environmental conditions. Light, water availability, balancing the need for CO2 uptake with the risk of water loss, transpiration. This process also helps create turgor pressure in cells, keeping the plant firm.
And photosynthesis happens in the chloroplasts.
Correct. Inside the chloroplasts, the thylakoids, stamped into grana, contain the chlorophyll and are the site of the light-dependent reactions, capturing light energy. The surrounding fluid, the stroma, is where the light-independent reactions, Calvin cycle, occur, using that captured energy to convert CO2 into glucose. And just like animals need minerals, plants also need essential mineral nutrients from the soil, absorbed by the roots. We divide them into macronutrients, needed in larger amounts, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur plus carbon, hydrogen, oxygen from air, water, and micronutrients, needed in tiny trace amounts, iron, manganese, zinc, copper, boron, molybdenum, chlorine, nickel. Each has specific roles in plant growth and metabolism.
Okay, we understand how organisms build themselves and get energy, but life isn't always easy. There's the constant threat of diseases that disrupt normal functioning. How do scientists generally categorize diseases?
Classifying diseases helps us understand how they spread and how to manage them. Some key categories. Communicable disease, an illness caused by an infectious agent, like bacteria, viruses, fungi, parasites, that can be transmitted from one person or animal to another, either directly or indirectly, for example, influenza, COVID-19, measles. Non-communicable disease, NCD, a disease that is not transmitted from person to person. These are often chronic conditions resulting from a combination of genetic, physiological, environmental, and behavioral factors. For example, heart disease, stroke, cancer, diabetes, chronic respiratory diseases. We also talk about duration and spread. Chronic disease, a condition that persists for a long time, often for life, and typically cannot be cured completely but can be managed. Example, diabetes, arthritis, heart disease. Then terms for outbreaks. An epidemic is when a disease affects a significantly larger number of people than usual within a specific population or geographic area over a specific period. Endemic means the disease is constantly present in a certain population or region at predictable levels, for example, malaria in parts of Africa. A pandemic is an epidemic that is spread over multiple countries or continents, usually affecting a large number of people globally. Example, 1918 flu, HIV/AIDS, COVID-19.
Let's focus on diseases caused by pathogens, microscopic invaders. Starting with bacteria. What makes them unique disease agents?
Bacteria are single-celled prokaryotes. They're living organisms that can reproduce independently. Many are harmless or even beneficial, but pathogenic bacteria cause disease by invading tissues or producing toxins. Importantly, most bacterial infections can be treated with antibiotics, drugs that target specific bacterial processes like building cell walls. However, antibiotic resistance is a major growing problem.
What are some major diseases caused by bacteria?
There are many, unfortunately. Some key examples: tuberculosis, TB, caused by Mycobacterium tuberculosis, primarily affects lungs, spread through air. Vaccine, BCG, pneumonia, inflammation of lungs, often caused by Streptococcus pneumonia or other bacteria. Cholera, caused by Vibrio cholerae. Severe diarrhea, dehydration from contaminated water, nicknamed Blue Death. Tetanus, lockjaw, caused by Clostridium tetani toxin, usually from wound contamination, causes severe muscle spasms. Typhoid fever, caused by Salmonella typhi from contaminated food water. High fever, systemic illness, houseflies can spread it. Leprosy, Hansen's disease, caused by Mycobacterium leprae, affects skin, nerves, respiratory tract. Diphtheria, serious infection of nose and throat. Gonorrhea, a common sexually transmitted disease, STD. Whooping cough, pertussis, caused by Bordetella pertussis, highly contagious respiratory illness. Meningitis, bacterial forms. Urinary tract infections, UTIs, are also common.
Okay, bacteria are treatable with antibiotics, usually. What about viruses? They're different, right? Not quite alive.
Exactly. Viruses are much smaller than bacteria and structurally simpler. They consist of genetic material, DNA or RNA, inside a protein coat, capsid. They're obligate intracellular parasites. They cannot reproduce or carry out metabolism on their own. They must infect a living host cell and hijack its machinery to replicate. Because they lack their own cellular machinery, antibiotics are completely ineffective against viruses. Treatment relies on antiviral drugs, which target specific viral processes if available, or the body's immune system, often supported by vaccines.
What are some major viral diseases?
Again, a long list affecting humans. Influenza flu and COVID-19, caused by SARS-CoV-2. Highly contagious respiratory viruses. COVID-19 is a zoonotic disease jumped from animals, HIVs, caused by the human immunodeficiency virus, HIV. A retrovirus, RNA genome, transmitted via blood-sexual contact. Attacks the immune system, leading to AIDS, tested via ELISA, hepatitis B, viral infection attacking the liver, transmitted via blood-body fluids, can cause chronic liver disease, cancer, measles, mumps, rubella, MMR. Contagious childhood illnesses preventable by vaccine. Chickenpox, varicella. Common viral illness causing itchy rash, vaccine available. Shingles is reactivation of the same virus. Smallpox, eradicated globally by vaccination, pioneered by Edward Jenner. A major historical killer. Polio, caused by poliovirus, can cause paralysis, largely controlled by vaccines. IPV Salk, OPV Savin. Rabies, transmitted by animal bites, dogs' baths. almost always fatal if symptoms appear, but preventable with post-exposure vaccination. Dengue fever and Zika virus, spread by Aedes mosquitoes. Dengue causes fever, pain, Zika linked to birth defects. Ebola virus disease. Severe hemorrhagic fever, high mortality, spread via direct contact with bodily fluids. Herpes simplex virus, HSV, causes cold sores, HSV1, and genital herpes, HSV2. Human papillomavirus, HPV, very common STI. Some strains cause genital warts, others linked to cancers, especially cervical cancer. Vaccine available, avian influenza, bird flu. Strains like H5N1 primarily affect birds but can jump to humans, raising pandemic concerns.
Bacteria viruses. What about other microscopic culprits like fungi and protozoa?
Yes, they cause diseases too. Fungi are eukaryotic organisms, like molds, yeasts. Many are harmless, but some cause infections, mycoses, often on skin or mucous membranes, but sometimes internally. Common examples: athlete's foot, ringworm, tinea infections, thrush, candida infection, aspergillosis, lung infection. Some types of scabies caused by mites can have secondary fungal infections. Fungal infections can also contribute to baldness or trigger asthma. Protozoa are single-celled eukaryotic organisms, often parasitic. They cause major diseases, especially in tropical regions. Malaria, caused by Plasmodium parasites transmitted by female Anopheles mosquitoes, causes cycles of fever, chills. Tested using VVAX test, among others. Traditionally treated with quinine from cinchona tree. Ronald Ross discovered mosquito transmission. World Malaria Day is April 25th. Amoebiasis, amoeba dysentery, caused by Entamoeba histolytica from contaminated water food, causes severe diarrhea, leishmaniasis, Kala azar, caused by leishmania parasites, transmitted by the sandflies, affects skin or internal organs. Sleeping sickness, African trypanosomiasis, caused by trypanosoma parasites, transmitted by the tsetse fly, affects nervous system.
So infectious agents are a huge category. But some diseases aren't caught. They're inherited genetic disorders.
Exactly. Genetic disorders result from abnormalities in genes or chromosomes. They can be inherited or arise from spontaneous mutations.
Yeah.
Some well-known examples: Down syndrome, caused by an extra copy of chromosome 21, trisomy 21, leads to characteristic features and developmental delays. Inherited blood disorders like thalassemia and sickle cell anemia affecting hemoglobin, color blindness, difficulty distinguishing certain colors, usually X-linked inheritance. Turner syndrome affects females, caused by missing or incomplete X chromosome. Albinism, lack of melanin pigment. Others include Angelman syndrome, neurological, Klinefelter syndrome, males with extra X chromosome, XXY. These highlight how crucial correct genetic information is.
And plants aren't immune either, right? They get sick too.
Definitely. Plant diseases cause massive agricultural losses. Bacteria cause things like bacterial blight and bacterial wilt. Viruses cause diseases like tobacco mosaic virus and barley yellow dwarf. Fungi are major culprits, causing late blight of potato, infamous Irish famine cause, rust of wheat, powdery mildew, etc. Plant pathology is a huge field.
Any other interesting disease-related facts to wrap this up, maybe about immunity or specific conditions?
Sure, a few quick ones. Specialized white blood cells called lymphocytes, specifically B cells, are responsible for producing antibodies, key proteins in adaptive immunity. Pasteurization, heating liquids like milk, reduces harmful bacteria, making it safer. Named after Louis Pasteur. SARS stands for Severe Acute Respiratory Syndrome, a viral illness that caused an outbreak in 2003. Environmental toxins. Radioactive strontium-90 can cause skin cancer. A-type disease from cadmium poisoning. Blackfoot disease from arsenic in water. Minamata disease from mercury poisoning. Nutritional deficiencies. Kwashiorkor from severe protein deficiency in children. Other conditions. Hyponatremia, low blood sodium. Glaucoma, eye condition damaging optic nerve, often due to high pressure. Trachoma, bacterial eye infection, leading cause of preventable blindness. Remember, retroviruses like HIV have RNA genomes and use reverse transcriptase. And houseflies can mechanically transmit pathogens like those causing typhoid. Health is complex.
Wow, okay. We have covered an absolutely incredible amount of ground today. A real deep dive from cells to ecosystems, reproduction to respiration, health to disease. It really highlights how interconnected everything is.
It truly does. You start pulling one thread in biology, and you find it connected to everything else.
Yeah.
The elegance and complexity are just staggering when you step back and look at it all.
Absolutely. As we kind of wrap up here, let's touch on a few more of those fascinating biological facts, oddities, and discoveries that just make you go, huh. Kind of like biological trivia, but often profound. Starting with some human body marvels.
Yeah, the human body is full of surprises.
Yeah.
For instance, did you know that brain cells, neurons, have the least regenerative power of almost any cell type in our body? Once they're gone, they're mostly gone for good.
So protect your brain cells.
Definitely. And linking to that, nerve cells are also the longest cells in the body. A single axon can run from your spinal cord to your big toe. And most neurons don't undergo cell division after birth, which is related to their limited regeneration. Your eye color. That's determined by the pigment, melanin, in your iris. The iris also controls your pupil size, regulating light. An adult human has 206 bones. The femur, thigh bone, is the longest and strongest, and the hardest substance in your body. Not bone, but tooth enamel. Incredible durability. We typically have 4 canine teeth as adults. The study of teeth is called odontology.
What about things we don't really need anymore? The vestigial organs?
Right, we have remnants of our evolutionary past. The tailbone, cossycetics, is a vestigial tail. Wisdom teeth were useful for our ancestors' tougher diet, but now often cause problems. A few quick stats. Normal adult respiration rate is about 14-18 breaths a minute. The liver is the largest gland. Normal blood pressure is around 1/10 of 80 millimillimeters She. Normal body temperature is about 37 degrees C or 98.7 degrees Fahrenheit.
And some food-related ones?
Sure. Cow's milk has a slightly yellow tinge due to carotene, related to vitamin A. The sweetest natural sugar is fructose, found in fruits. The bacteria that turns milk into curd yogurt is typically Lactobacillus acidophilus, and the dietary fiber that keeps us regular is largely cellulose from plant cell walls, which we can't digest.
Cool facts. Now shifting to some of the giants whose discoveries shaped biology.
Absolutely. We owe so much to these pioneers. Parthasarathi Iyengar is revered as the father of Indian phycology, study of algae. Charles Darwin gave us the theory of evolution by natural selection, 1859, fundamentally changing how we view life's diversity. Gregor Mendel, through his pea plant experiments around 1865, rediscovered later, established the basic principles of heredity and genetic inheritance. Father of genetics. In cell biology, George Palade discovered ribosomes, 1955, and Christian de Duve discovered lysosomes, also 1955. Nobel Prizes followed. Medical breakthroughs. Frederick Banting and Charles Best discovered insulin, 1921, saving countless lives from diabetes. Structural biology. G.N. Ramachandran elucidated the triple helical structure of collagen. Linus Pauling and Robert Corey determined the fundamental alpha helix and beta sheet structures of proteins. E. Gorder and F. Grandel discovered the lipid bilayer structure of cell membranes, 1925.
And who came up with some of these terms we use all the time?
Good question. The term gene was coined by Wilhelm Johansen, hormones by E. H. Starling, in ecology, pyramid of numbers by Charles Elton, ecology itself by Ernest Haeckel, and the crucial concept of the ecosystem by Arthur Tansley. Karl Landsteiner discovered the ABO blood group system, leading to safe transfusions, Nobel Prize winner. O-negative is the universal donor type, and EEG, electroencephalogram, measures the brain's electrical activity. And who could forget Dolly the sheep, the first cloned mammal, 1996.
What an amazing legacy of discovery. Finally, a few more plant oddities and facts.
Plants are always full of wonders. Xerophytes are plants adapted to survive in very dry conditions with little water, like cacti. Bonsai is the Japanese art of growing miniaturized trees in pots. Creepers are plants with weak stems that spread along the ground. Carrots are orange due to carotene. The petiole is the stalk attaching a leaf blade to the stem. A node is the point on a stem where a leaf arises. The smallest flowering plant is Wolffia. Chlorophyll contains the element magnesium at its core. In an apple, the part we eat is mostly swollen thalamus, the false fruit. Red rot of sugarcane is a fungal disease caused by Colototrichum falcatum. The sting from a nettle plant is caused mainly by methanoic acid. Xylem cells are waterproofed and strengthened by lignin. Heliophytes love full sun. Psiophytes prefer shade. Hydrocolloids, like agar alginates, are water-holding substances from marine algae. Algae perform at least half of the Earth's total CO2 fixation through photosynthesis, hugely important globally. Chlorella is a protein-rich green algae, sometimes called space algae. Laminaria is a brown algae known as kelp, storing food as laminar in mannitol. In mosses and ferns, bryophytes, pteridophytes, the antheridium produces sperm, and they need water for the sperm to swim to the egg for fertilization. Phytoplankton are microscopic photosynthetic organisms forming the base of most aquatic food webs. And the principle of complementarity, base pairing rules, ATGC, governs how DNA is transcribed into RNA.
Wow. Okay, we have truly journeyed across the biological landscape today, from the tiniest cellular machines and genetic codes up to the grand classification of kingdoms, the complex systems of digestion and respiration, the miracle of reproduction, the challenges of disease, and some just fascinating facts. It really underscores how these fundamental biological principles govern everything.
It's been quite the deep dive, and what really strikes me, reflecting on all this, is the sheer elegance and sometimes the unexpected simplicity behind life's solutions. You know, how basic physics like diffusion powers gas exchange, or how adding just one extra layer of the mesoderm during development opened the door to such incredible animal complexity. It's truly awe-inspiring.
That's a really powerful takeaway, and thinking about the bigger picture, it raises a crucial question for all of us, doesn't it? Considering this vast diversity, this interconnectedness of all life that we've explored, how might really understanding these basic biological principles empower us? Empower us to better protect our fragile ecosystems, maybe innovate more sustainably, perhaps even improve human health in the long run?
Absolutely. It prompts a deeper reflection, I think, on how our own everyday actions, what we choose to eat, how we impact our environment, even just understanding our own bodies, are all deeply interwoven with these biological processes we've discussed. Knowing the why behind it all can be quite profound.
So the invitation to you, our listeners, is to let this knowledge change your perspective. Think about the biology happening within you and around you right now. Keep asking questions, keep observing the living world, and stay curious about this incredible dynamic blueprint that makes life in all its forms possible. It's constantly at work, often without us even noticing.